Good morning.
Good morning, sir.
Okay. Uh, Today we have 21 people on board. Fine. So let's begin. So yesterday we started with the topic that is the quaternary structure. So if someone asks you about the definition about the quaternary structure, you can say it is simply the subunit interactions. Okay. So maybe a given protein or a given polypeptide will be first converted into its three-dimensional structure or a tertiary structure, which we can call as the subunit. Then again, these subunits, they will interact with each other and they will lead to the structure that is the quaternary structure. Fine. So simply, it could be the continuation of your tertiary structure, whereby these two or more than two tertiary called subunits interact with each other, forming the quaternary structure of your protein. Now, obviously, if the interaction has to take place, there has to be some or the other kind of the complementation between the subunits, right? And that complementation will help you to form the quaternary structure of the protein. Now, when the quaternary structure of the protein is formed, always there is a symmetry which is maintained. Okay, so this symmetry could be with respect to that means if you try to break that particular protein, you may get an identical horizontal symmetry. So with few of the examples or the diagrams which I have showed you, so it could be isologous, it could be heterologous, or the homologous type of the uh, interactions between the subunits. And obviously the sub interactions which are taking place, or it could be the hydrophobic type of the interactions, right? So to continue with this particular topic, I will be presenting the screen so that we'll try to get more insights into that how this quaternary structure is being formed. Is the screen visible?
Yes, sir.
Okay, fine. So we have discussed about these topics, that what happens when it is the homologous type of the interactions, when it is heterologous. So yeah, so in case of the homologous, you get that particular type of the symmetry. But when it comes to the heterologous, we have seen that either you may get an open-ended structure or you may get an close-ended structure, but it is cyclic in nature. Now, when such type of interaction is there, that is interaction between the non-identical subunits, the symmetry of that particular proteins could not be maintained. Okay, and that becomes a problem. But still, those interactions are happening. So, is it that only few of the proteins are having symmetry and the other proteins are not having symmetry? No, all of them, they have the symmetry. But now here, the symmetry could not be with respect to the horizontal or a vertical type of the symmetry which I told you, but it has to be an rotational symmetry, right? And this is what the scientists they came to know when they tried to study the what you can say the larger proteins. Okay, means specifically the virus code proteins. When they were studying it, they found that the vertical or the horizontal type of the symmetry could not exist, but there is a symmetry which we can call it as an rotational symmetry which is present into the quaternary structures of the proteins. Fine. And there always a particular structure of the protein will try to remain into an low symmetry structure, because that low symmetry structure is a stable structure. Why? Because the energy associated with that particular structure is very, very low. Yeah. So we'll be talking about the cyclic symmetry. So now we can classify or divide these particular proteins based on their cyclic symmetry, that how many types of cyclic symmetries are available in a given protein. Fine. So what does it say? That here the subunits, they are arranged around a single rotation axis. Okay, so at one particular point in the center you may consider it as an axis of rotation, and around that axis of rotation you can easily rotate it. Now whenever you are trying to rotate this subunit, okay, by any angle, always you will be getting the same type of the structure. So there is no variation or deviation in the structure. Okay, so if you have more than two subunits, we refer to it as what, the two-fold rotation axis. Okay, and this rotation axis can rotate the quaternary structure at 180 degrees. Always you'll be getting the identical type of the structure to that of the original one. Maybe I'll show you the diagram so you'll maybe be able to understand it. Yeah. So looking at these three diagrams, if you see here, the first diagram shows angle of rotation. And if I rotate this at by 180 degree, now there are only two subunits, so I can rotate it at 180 degree. So simply what will happen, this subunit will come on this side and this subunit will come on this side. Okay, so if you look at the structure, you will see that this subunit is having more than two subunits. So now if your protein is having more than two subunits, okay, so in that case only the variation could be that the rotation what you are trying to cause, it could be decrease. Say suppose here you have the three subunits. So simply the 360 divided by 3, so it may come up to 120. Okay, so if you have these three subunits are arranged in such a way that when they rotated at this axis of rotation by an angle of 120, simply 120. So what will happen? Just imagine that this subunit will come here, this will come here, this will come here. Okay, so even if you are rotating at 120, you get the identical type of the structure as that of the original protein structure. Right. The same way, if you go on increasing the subunits, okay, only the angle of rotation may change. So here now you have 5. So just you can calculate that how much angle of rotation could be there here. So if you rotate it by around something 60 degree, 40 degree, etc., etc., you get the identical type of the structure. So such type of symmetries are observed. These are called as what? The cyclic symmetries. Okay, and based on these cyclic symmetries, you have the different classification of the proteins which has been done for the quaternary structure of the protein. Right. So as I told you, in this case it could be 120. In this case it could be around what it will be, किती होईल याच्यात 5 आहेत, 360 भागिले 5 करा बरं, 5 7 35, so something around 72 degrees. If you rotate it, you will get the same type of the structure. Fine, I think you got the idea now. Now as the number of subunits it goes on increasing, obviously you may not have only that one fold of rotational symmetry. So in that case you have the dihedral symmetry. Okay, so now this dihedral symmetry is an additional symmetry which could be obtained, okay, along with the rotation axis. Okay, so this dihedral symmetry is an additional symmetry which could be obtained along with the n-fold rotation axis, which is perpendicular to the another n-fold rotation axis. So just I'll show you the diagram so you'll be able to understand. Example could be your concavalin A and insulin. See, now this is the axis of rotation. Now if I rotate in this particular direction, you get the identical subunit. axis of rotation on this side where you can rotate it like this, right? So same way you can see here where there are 8 subunits. The same way you can see here there are 6 subunits. So so such type of you can see the protein structures in which the number of subunits could be more, you may have the dihedral symmetries. Okay, and this in this dihedral symmetry proteins you have these axis of rotations which are perpendicular to each other, right? Now, as I told you that based on these symmetries you can classify. So you have the higher symmetry groups also, like you might have heard about the tetrahedral, it could be the octahedral, it could be icosahedral symmetries, etc., etc. Fine. Again, here these symmetries, the higher symmetry groups, where I told you that it could be that you have the number of subunits which could be more than 10 or 12. Okay, so one of the example which I showed you that there were around 120 subunits in case of the code protein of the viruses. So in those case, the structures could look something like this. I'll not read out. You can get it afterwards. Yeah. So just look at this particular. Okay, and this specific arrangement of the subunits and their symmetries is giving the specific shape to that particular protein. So now you can imagine that where these proteins could be present. So this is the tetrahedral, this is the octahedral, this is the icosahedral, where you get that exactly the virus code protein which could be seen here in case of the icosahedral symmetry. Right? So this is all about the quaternary structure, where it is simply the interactions between the subunits, and it depends upon that how the subunits they interact with each other, whether they are identical, not identical, whether they are having the rotational symmetry, or it is having a dihedral symmetry, etc., etc. So I think you got that particular thing about the quaternary structure of the protein. Now the next question comes, that what are the forces which are driving this quaternary association? Okay, obviously the forces could be the same, that is the non-covalent interactions. Fine. So understanding those particular points, or those basic points what we already covered, there are few points which I am trying to tell you here. So this particular structures of the quaternary structures has been evaluated for very few proteins. By analyzing those particular proteins, what are the observations which helps us to understand that what kind of forces or what amount of energy is required to stabilize the such a huge amount of protein. So they have calculated the dissociation constant, and they found that the dissociation constant for these subunit interactions or the associations, it range from 10 raised to 8 to 10 raised to 16. Okay, and this dissociation constant, it relates to the free energy change. Okay, so the free energy change in this case, it could be around something 50 to 100 kilojoules per mole at 37 degrees Celsius. Obviously this, it is going to change if we change the temperature. So we are talking at the 37 degrees Celsius, which is the ambient temperature of that of the cell. Okay, so the energy required for the association could be in the range of 50 to 100 kilojoules per mole. Now this dimerization or the association between the subunits, as we have seen that it could be accompanied by both the favorable as well as the unfavorable energy changes. Okay, means if there are two a repulsion. Okay, but if they are identical, there could be an attraction, and you may get the place. Now here the favorable interactions are what? All the non-covalent interactions what we have discussed. So it could be the van der Waals interaction, the hydrogen bonds, ionic bonds, hydrophobic interactions, etc., etc. Okay. Now here when this subunit interaction is taking place, obviously what is going to happen? The entropy is going to decrease. Okay, because now these subunits, they are interacting with each other, they are forming one particular structure. So here there is a considerable loss of the entropy whenever there is an interaction which is taking place between the subunits. Now in a given protein, if you have more than two subunits, okay, so it could be three, four, and in within that particular structure, say so one example which I am trying to explain here, that you have a protein which is having three subunits. Now out of these three subunits, if there are two subunits which are moving, so obviously there will be three the translational degrees of freedom. The degree of freedom means the rotation, the axis of rotation, that could be lost for that particular subunit, because it will be constrained. हे बघा, if they have to move, they have to move as a three subunit structure. But if you try to move only two subunits, it will, there will be a constraint by the third subunit, because the third subunit will not allow it to move. So whenever you are trying to imagine that if there is a change or a movement which is taking place into the protein, it has to be the whole structure which is undergoing that particular change. Okay. So this subunit association, it restricts the free movement of the residues at the subunit interface. I think these sentences, what I have tried to write here, they are the self-explanatory sentences. So if you read them, you will be able to understand what I'm trying to say here. Then due to this restriction, means that could be an unfavorable type of the association, and this unfavorable energy range in the range of 80 to 120 kilojoules per mole, obviously at 25 to 37 degrees Celsius, right? So that is compensated by the favorable energy of association, right? So you can see here, thus to achieve the stability, the dimerization of the two subunits must involve approximately 130 to 220 kilojoules per mole of favorable interactions, right? So that many amount of the favorable interactions are taking place, so that this unfavorable energy, which is not allowing the subunits to come together, that can be removed, and you get the structure which is formed. Okay. And here the favorable energy which is helping to overcome this unfavorable energy of association, here the major contribution is of the uh, van der Waal interactions or the hydrophobic interactions. Although they are not a true type of interaction, but they are many these kind of interactions which is giving that stability to the protein, or providing that much amount of free energy, so that the favorable association can take place and you get a very stable structure. Okay. So this van der Waal interaction itself, it could account for around 150 to 200 kilojoules per mole. Okay. So if that much amount of energy is associated with the van der Waal interactions, that energy is more than sufficient to overcome this unfavorable energy of association, right? And that is why we can have the proteins into their quaternary structure. Okay. So I think you got the point what I am trying to explain here about the favorable and the unfavorable interactions. Now, in addition to these non-covalent interactions, in some of the proteins, we may have some additional factor which is involved or which is responsible for forming the quaternary structure of the proteins. And here again the same example will come, that is the disulfide bonds which are formed between the different subunits. And again the best example which can be quoted here is a immunoglobulin. So all the antibodies, they are 22 tetramers composed of two heavy chains and two relatively light chains. I think now you might have studied the structure of the immunoglobulin. Okay, so that many subunits are there, and all those subunits, they are held together by the disulfide bonds. Okay, so in addition to the intra-subunit disulfide bonds, you also have the inter-subunit disulfide bonds. So you might have seen that there is an interaction between within the light chain and within the heavy chain. You also have the interaction between the light chain and the heavy chain. Okay, so both the type, that is intra as well as the inter type of the disulfide bridges are there, which will hold together all the subunits of the light chain and the heavy chain, so that you get the typical structure of the immunoglobulin, which can have its structure. Okay, so you have the light chain, heavy chain. So you can see that there is an intra, that within the light chain and heavy chain you have the disulfide bonds, and also you have the disulfide bond between these two subunits, right? So I think this example will help you to understand about the disulfide bond. So based on this information, you have the different modes and models for the quaternary structures. Okay, here I'm not going in too much of details. I'm just trying to give you an idea about the quaternary structure. So when you talk about the models of the quaternary structure, the structure, it will be simple if that quaternary structure is made up of the similar subunits, I mean the homomers. Okay, homomultimers you can say in that case, right? But it becomes complicated or difficult to study if it is made up of different subunits, that is hetero multimers. So one example which I have brought here, that is the hemerythrin. Hemerythrin, it is an oxygen carrying protein in marine vertebrates. Okay, and it has a very compact arrangement of four antiparallel helices forming subunit. So like this hemerythrin, it can exist in the different mers in the different organisms. Okay, but still it can carry out its function. So for that obvious reason, I have put this as an example here. So it can form the dimers, trimers, tetramers, or the octamers, or even higher types of the aggregates which are found for this hemerythrin. See here. So this is a simple subunit which could be present in one of the organisms. In some organism, you may have this tetramer, means no, this is not a tetramer, but this is an octamer. Means like this, 8 subunits they are interacting with each other, and you get the whole protein which could be obtained. Now here in this case, you can see that there are 3 subunits which are attached to each other, and you get the uh, function. The function remains the same. Only thing is that it can have variation into it number of subunits, and based on that you can now imagine that that much amount of oxygen can be uh, captured or made available to that particular organism. Fine. So you can see here that in case although it will be difficult to read the names, so it is thermisty dostericola. So in this particular organism, you have it in a monomer form. In a Goldi, you have it in octameric form. In a Siphonosoma, you have it in a trimeric form. Okay, so this is just to give you an idea that it can exist in the different forms. Okay, so the above example which I told you, it is an example of the uh, identical type of the subunits. Okay, but you may have some uh, different type of the subunits which are involved. So in that case, you can imagine that the uh, associations or the interactions will be a bit complicated type of interactions. So, in case of these multimeric proteins with more than one kind of subunit, often it display different affinities between the different pairs of the subunits, and there are different interfaces which are involved into the interactions. Okay, so the example again here you can quote is the hemoglobin. So hemoglobin, if you know, it consists of the 4 subunits. So there are 2 alpha subunits, there are 2 beta subunits. So just imagine that the that is the 2 alpha subunits will interact with each other first, then the 2 beta will interact with each other, and then they will form some surfaces between the 2 alphas and the 2 betas, and those surfaces will be having the complementation for each other, so that you get the overall structure of the hemoglobin which could be obtained, right? So that type of association is there between the different type of the subunits, and because of this particular variation into the association or the interactions in the hemoglobin structure, if you are trying to break the hemoglobin structure, you may require some strong chemicals which are strong denaturants, so that they can dissociate your hemoglobin into the individual subunits. Fine. So attempts has been made, and we are successful in separating the different subunit, that is the alpha subunit as well as the beta subunits. And there is a wide amount of study which has been done into this particular type of the proteins. Fine. So maybe that is more than sufficient to understand about the quaternary structure. I have tried to bring about the topic. I have tried to explain you a few glimpses about the quaternary structure, and if you can understand up to this particular point, it is more than sufficient for uh, this particular subject. Okay, in the further classes, maybe during your proteomic studies or bioinformatics studies, you may go beyond this, but at that time you may require all these basic things which I have told you up till now. Right. So that was all about the hierarchy between the protein structure. So from my side, I have tried to explain the whole process of the uh, protein folding, that how does this protein folding it takes place, and what are the different hierarchical stages which are involved when a particular polypeptide is being synthesized and it is undergoing the process of folding. Right. So as a whole, if you try to understand, what we have seen up till now is that the information which is required for the folding of a protein, it is contained into the primary structure. So for that, we have seen what is a primary structure. It is simply a sequential arrangement of the amino acids when a polypeptide is being synthesized, and that sequence is going to decide that what type of structure this protein is going to form. Okay, so you can imagine, or you can say that it is the first, you have the primary structure which is being obtained. Okay, but when this primary structure is being synthesized, at the same time you can see that there are some local conformation changes which are taking place, and these local conformation changes, it is leading to some it is leading to two types of specific secondary structures what we have seen. So they are nothing but the alpha helices and the beta sheets. Now once the secondary structures has been found or formed, what happens, they again start interacting with each other, and they may lead to something called as an super secondary structures. Okay, so it could be an interaction between two alpha helices, it could be an interaction between two beta sheets, or it could be an interaction between a helix and a sheet. Okay, and these interactions between the different secondary structures, they lead to the formation of the motifs, which can be further again interacting and forming the domains. Okay, so whenever this motif and the domains which are formed, means it is maximizing, it is trying to maximize the number of interactions. Okay, and that automatically is converted into the three-dimensional structure of the protein, and that we refer to as what, the tertiary structure. So whenever you are trying to refer about the tertiary structure, it is simply the maximizing the number of possible non-covalent interactions, so that you get a three-dimensional structure of the protein which is being obtained. Right? And in few of them, you may have a covalent interaction, and that is the disulfide bond. Okay. Now once the tertiary structure or the three-dimensional structure has been obtained, you can call it as what, a subunit. So if in a given protein which has to go up to the quaternary structure, you may have that many subunits which are formed, and in further stage what will happen, that these subunits will again start interacting with each other and forming the quaternary structure of the protein. Now here, it is not necessary that each and every protein what we have into the cell will be going up to the quaternary structure. So it will depend upon the function of the protein. So as I had been telling you, that some of the proteins, their folding may end up at the secondary structure itself. That is simply, it will be the formation of the alpha helical structure or the sheet structure. And the best example in that case could be your fibrous protein, because fibrous protein, they are always linear, so their folding ends up at the secondary structure. Okay, while in some of the proteins, or in majority of the proteins, where I say that the globular proteins, okay, so these globular proteins may result into the formation of the motifs, the domains, and the tertiary structure. Okay, and when you're talking about the globular proteins, so along with this secondary structure, you have some small structures which are formed, which we have studied. They can be called as what? The turns, the loops, the bulges. So these small structures, that is the turns, loops, or the bulges, they help in reorienting the direction of that particular polypeptide. So that helps us to fold the polypeptide so that a globular protein can be obtained. So majority of the protein, they may end up into their protein folding at the tertiary structure level, while in very few type of the proteins, like few examples what we have seen, it could be the hemoglobin, hemerythrin, which I told you, or basically the coat proteins of the viruses. In that case, you may see that those proteins, they end up with their protein folding up to the quaternary structures. Fine. But when this protein folding is done, as I had been telling you again, that the nature has selected the proteins in such a way that it will not go into the too complicated structures. Even if there are turns, loops which are present, or the folding is present into the protein, you may not have the complicated type of the folding. Okay, and you may not have majority of the proteins, which are the larger proteins, having all these type of the complicated structures. Right. So this is what you are supposed to understand or to learn when you are talking about the protein folding. And whenever you are talking about this protein folding, basically it is the non-covalent interactions which are playing a very important role. Okay, so here we can make a statement that in comparison with the covalent bond, लक्षात घ्या, in comparison to the covalent bond, the hydrophobic interactions or the van der Waal interactions are much more stronger, which is giving the stability to the protein structure. Okay, although these are the weaker bonds, or these are they are the imaginary bond, but still these bonds, they are in a huge number, or they are in a very large number, and that large number type of that large number of these interactions is making this structure a very stable and a strong structure. Okay, so that was all about the protein folding, which I was supposed to teach you here. Maybe if you look at the syllabus, the points which are present there, I have tried to explain in that particular way. When I was talking about the different models, that how does the protein folding it takes place, so there I have given you idea about the different factors which should be taken into consideration when you're talking about the protein folding. Then there are different models, that is the funnel model which I told you. Then some other models were there, the names, you can refer those particular names. But what does this model suggest? That always this extended conformation of the polypeptide is converted into the folded, that is the native conformation of the protein. And when this change is taking place from the extended to the native conformation, you may have many intermediates which are formed, and these intermediates, they interact and they form the native structure. Right. So that was all from my side. Any questions, any doubts, if you have in this whole process which I have tried to explain, you can raise some questions. Okay, either you can put it as a message into the Google Classroom. Okay, see what I'm trying to say, that you put it into the groups only. so that even if someone else is having that particular problem, I will be able to justify the answer in that case. Fine. So that was all about the protein folding. Yes, any doubts, any questions? Yeah, Nupur?
So, which
Yeah.
book we should refer for forces stabilizing tertiary structure? It's not there in Leninger, actually.
The Garrett and Grassing. Hmm? So that is the book in which
Okay,
the
sir.
diagrams what I have taken, it is from those that particular book. Still, if you don't get, you just let me know. I'll give you some other books also. Then that.
Okay, sir.
Yeah. Komal, Sakshi, have you understood the whole process?
Yes, sir.
Okay. Tejal,
Yes, sir.
sure. नुसतं हो म्हणू नका. It's not for my satisfaction. It should be your satisfaction. अवधूत.
Yes, sir.
हम्म. अरे नुसतं येस सर काय म्हणता रे, काहीतरी बोला ना. काहीतरी तुमचे